Hey folks, in this video I'd like to take a look at the differences in the types of nucleic acids, primarily DNA versus RNA. You probably all know this, but let's just define. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So I'm going to start out with the structure of RNA first. And so if we were to draw out a simple building block of RNA, we're going to draw a ribose sugar in a Howarth projection, like this, and so it's in the cyclic form. And so I'm going to darken in these bonds here. And I'm going to put a phosphate here on this position. And on this position, this is where the base would attach. So in RNA, we'll see there are 4 different nitrogenous bases that can attach. So as you may recall, ribose is a 5-carbon sugar. Let's number the carbons. Now when you're talking about nucleic acids, we often add a little 1' to the numbering just to show that these are part of nucleic acids. So this first carbon, which would be the anomeric carbon, that's 1'. This is 2', 3', 4', and then the one that's outside the ring would be called 5'. So this is called a 5' phosphate. And this entire structure that contains a phosphate, the ribose sugar, and a base is called a nucleotide. If it just had the alcohol, so if I did not have a phosphate on here and just had an OH at 5', that's called a nucleoside. One way to helpful maybe remember that is that there's a detergent you might have heard of called Tide. Detergents also have phosphates in them. So a nucleotide has the phosphate, whereas a nucleoside does not. It would just have the OH here. So this generalized structure then would contain possibly 4 different bases. So RNA is going to contain the following bases: A, U, G, and C. Okay, so envision now we have this base called A, which stands for adenine. That's going to be attached here. That would be the nucleotide called adenosine. Next one, if I had U or uracil attached, that would be uridine. G attached would be guanidine. C would be cytosine. Cytidine. I'll draw the structures of these bases out in just a little bit. All right, so that's the generalized structure of RNA. So we have this sugar ring of ribose, a base attached, and then a phosphate. And I should say this, what I've drawn here, the nucleotide, is the individual building block of RNA. An actual RNA molecule is going to be a long polymer of multiple of these connected together between the 5' position and the 3' position. We'll get to that in just a second. So these are the RNA nucleotides. If we wanted to talk about DNA, the only difference that's going to really exist is this position right here. The 2' position in DNA will not have this OH group here. It's just going to have a hydrogen. So let's redraw this sugar. So everything else here is pretty much going to be the same. I'll go ahead and put my phosphate on. And at the 2' position, instead of putting an OH, I'm going to put a hydrogen there. And then our bases would be attached here. So let me darken in the ring here just a little bit. So this is, again, 1', 2', 3', 4', and 5'. So just like before, if I have the phosphate on, that's called a nucleotide. But in this case, this is now going to be called a deoxynucleotide, or D, the lowercase d for deoxygenated. So notice the difference again. So this is often called a 2' deoxy sugar because the 2 position does not have the OH group on there. And these would then ultimately be the building blocks of DNA. Again, if I didn't have the phosphate on there, that would be called a nucleoside. And both of these would be deoxy in front of them. All right, so this would be the structure of the DNA building blocks, the monomers that ultimately will get linked together to make DNA. And then lastly, what we should just say is that there is one other major difference between the DNA and RNA. The bases are slightly different. Instead of uracil, DNA is going to have A, T, G, and C. So the main difference in terms of the bases is that in RNA you have U or uracil, and DNA you have T or thymine. So what I'd like to talk about right now is the structures of the bases that would be attached to either DNA or RNA. And there are two main classes of the bases. The first are called the purines, and they're based off the general core structure that looks like this. This is the organic nitrogenous base called purine. It's got a 6-membered ring and a 5-membered ring with 4 nitrogens in it. And essentially in the nitrogenous bases that occur in DNA and RNA, these purine rings are now going to be functionalized with some other things attached to it. So if we just do the following modifications, what I'm going to do is I'm going to put an amino group up here at the top of the 6-membered ring, and I'm going to just put this squiggly line here at this position. This is the position, the nitrogen that would be attached to the sugar ring of our nitrogenous, nucleotide. This would be an adenine, or A, the base A. Conversely, if I did another slight modification of this, where I now have this structure, this is guanine, or G. So notice that there's a different functionalization on the same purine core, which is this, which gives rise to these two bases, A and G. So those are the so-called purine bases that you'd find in either DNA or RNA. Now the other types of bases are called pyrimidines, and they're based off of this smaller nitrogenous organic base called pyrimidine. And again, we're going to functionalize this core structure a little bit differently. So let me start off with this structure. And this structure here that has a methyl group, 2 C double bond O's, and an amide group double bond here in this position, this is thymine, or T. So that's the base that we would abbreviate as T. Next, we would have C, or cytosine, or C. And notice it's different in structure. It has an amino group up here instead of the C double bond O. It also is lacking the methyl group. And then lastly, uracil. It's going to have a structure that's actually sort of similar to T, thymine, but notice it doesn't have the methyl group. It has a hydrogen. This is uracil, or U. And again, U is only going to be found in the ribonucleotides, or in RNA. So envision now these bases are going to be now attached to the anomeric carbon of our sugar ring. So for example, let me just over here draw one of these. I'll draw the phosphate that has a T attached to it, a thymidine. Now I'm going to abbreviate my phosphate as a P with a circle in it. Some professors think that's okay, that's fine. I also kind of like you to draw the C double bond O or the P double bond O with the O minuses, but that's okay. We'll just abbreviate it like that. So if I wanted to envision taking this thymine now and attaching it to the sugar ring, it would look like this. There's my CH3. So the key bond that's formed, the glycosidic bond to carbon 1', is a CN bond. And notice it's in the beta orientation. So the nucleotides are always found so that the bases are attached to the top face of that ribose type ring. So I've drawn the ribonucleotide. These also come in all the various flavors, but instead of having an OH, I could have a hydrogen here. The only one that would not have that would be the uracil version, since that's only going to be incorporated into RNA. All right, so that sort of takes care of the basic building blocks of RNA and DNA. In other videos, we're going to talk a little bit more about how these form long polymers, and then ultimately how these lead to the ability to transmit genetic information. But this, again, just covers the basic building blocks. I hope that helps.